So question number 12, Vishwa is saying CDE. What is your guess? BCDE is what? Doctor, your name, doctor? Huh? I am a presbyopic, presacutic, andropausing, geriatric teacher. You must speak little loudly, doctor. Huh? Spell. Thumbkeen. Huh? Okay. Thumbkeen's answer is very good. Srinivas and Raghav are also proposing ACE. No, doctor. You should be careful. Ovary is attached to the uterus by ovarian ligament is true. Uterine artery supplies medial 2/3 of the tube is also true. Is the ampulla longest or widest? Widest. Huh? So uterine tube length कितना होता है डॉक्टर? 20 centimeters is 2 tubes मिला के 10 centimeter in length and 1 centimeter in diameter and situated in mesosalpinx is what you have to basically remember. Yeah? Option B. Where is B? No, it is medial to lateral or not in that order. Huh? Lateral to medial is in that order. SIADH, syndrome of inappropriate ADH secretion may. How will a patient present clinically? Patient presents with altered sensorium. When you evaluate the patient, you will discover there is hyponatremia. So why hyponatremia is there? You will check. Generally, if hyponatremia is there, common cause क्या होता है? Decreased intake. In the home, they will eat avakai, magai, nimmakai, musirkai, all types of salted pickles. When you are admitted in an intensive care unit, that privilege is not there, and we are pushing diuretics into the patient, and he is losing the sodium into the urine. Due to all these various causes, sodium loss into urine generally lead to hyponatremia in intensive care unit patient. But there is no history of diuretics, there is no liver disease, no adrenal disease, no lung disease, no thyroid disease, no edema. Patient is euvolemic, not dehydrated or edematous. In a euvolemic individual, if there is a hyponatremia, then it is called inappropriate ADH secretion. When ADH is inappropriately secreted, what will it do? It lead to retention of water. That retained water, what will it do to the plasma? Plasma become more in volume. And what will that do? It will stretch the atria, and that releases the atrial natriuretic peptide, and that lead to natriuresis. So how will be urinary sodium? Increased. So urinary sodium typically is increased, serum sodium is typically decreased, and since water is retained, serum osmolarity is low. Since water is not going into urine, urine osmolarity is high. So that combination is called SIADH, is what you need to remember. Okay, doctor. Maybe this description is too short, but in the video library of anatomy2medicine.com, you have the complete review, 40-minute review lecture on SIADH. You can go back home and review that. Okay, doc. Now, let's go to the next question. For the cyanosis to appear, how much of hemoglobin which is deoxygenated must be there, or a reduced HB level should be how much? The total hemoglobin should be 10.9, it will occur if total hemoglobin, and if the reduced hemoglobin is 6.1 also it will occur. So what is the basic criteria for the central cyanosis to occur? Minimum criteria, reduced hemoglobin should be at least 5 milligram. So it should be more than 5 milligram. Out of this, which is more than 5 milligram, doctor? 6.1 is, 6.1 is more than 5, and total hemoglobin is how much? 10.9. हो सकता 8.9 6.1 हो सकता, मगर 10.9, 4.1 is less than 5. Anything less than 5 of reduced hemoglobin does not lead to development of cyanosis, is what you have to basically remember. I told you na, secondary messengers is one of the favorite question of the examiner. Now the simplest question asked by the examiner is, what are secondary messengers out of all this? Cyclic AMP and calcium. Maybe in the entire history of PGI, easiest question in physiology. Okay, don't expect examiner will be that easy. Parathyroid versus vitamin D. Let me tell you, how does vitamin D metabolize, how does it act, what is the effect on GIT, what is the effect on kidney. These are all the issues. Vitamin D, parathyroid, calcitonin. 100% you should be sure. You must bookmark and prepare for the tomorrow's exam. The role of the parathyroid is to stimulate the 125 vitamin D formation. True. If parathyroid is excessive, it leads to resorption of the calcium from the bone and the bone become fibrous and cystic. That's the reason whenever excessive parathyroid is there, what is that entity called? Osteitis fibrosa cystica is the name given, because it will resorb all the matrix from the bone. When will high parathyroid occur in an individual? Common condition, tomorrow you are going to be nephrologist. Chronic renal failure may, if there is no vitamin D, calcium absorption is low. That hypercalcemia stimulates parathyroid release, and release of parathyroid resorbs the bone, and that lead to fibrous osteitis fibrosa cystica is what you have to basically remember. So the parathyroid causes the calcium resorption from the bone. It will decrease the loss of calcium into urine. It will cause increased absorption of the calcium into the intestine. But the third effect of the parathyroid is not direct effect of parathyroid. Parathyroid causes increased vitamin D production, and that vitamin D in turn will increase the absorption of the calcium in the intestine, is what you have to ultimately remember, doctor. That's what I want to underscore to all of you. And this is also another favorite question, please say. Another favorite question, doctor. Will all the effects are direct effects of parathyroid, but not which effect, is a favorite question of the examiner. Now, oxygen dissociation curve. What is accept about oxygen dissociation curve? Simple question. Even examiner is not asking what is right shift and left shift. But ODC, right shift, left shift is a favorite question of the examiner. In any PGI exam you go, right? So 2-3 dBg, CO2 pressure, acidosis, temperature, anything can ultimately affect. Which are the polar amino acids out of all these things, doctor? Basically, two challenges in amino acid topic. First chapter, in fact, first class when we join medical college is biochemistry, amino acids. But those days it doesn't look difficult. Why? Organic chemistry, left and right we prepared to get a seat into medical college. First year only 20 structures, IUPAC nomenclature and diagrams means फटाफट हो जाता, right? So slowly we will keep losing our neurochemical reserves. By the time we come to final year, even paracetamol dosage also looks painful, right? It should not be. So two challenges क्या होता है amino acid topic में? Essential, non-essential, polar and non-polar, glucogenic and ketogenic. ये तीन lists आपके pocket में लगा के जाना है. Definitely they will ask you a question. Non-polar are hydrophobic. Alanine, leucine, isoleucine, valine, methionine, phenylalanine, tryptophan and proline. Polar are hydrophilic. Non-polar are hydrophobic. Right? Then polar amino acids with positive R group, lysine, arginine. With negative R group, aspartate and glutamic acid. So all these possibilities you must basically remember. What are irreversible steps of glycolysis? Once more you can see PGI aims ABPG, TNPG, TGPG. 8 cycles: glycolysis, glycogenesis, genolysis, beta oxidation of fatty acids and synthesis, purine, pyrimidine metabolism, and cholesterol ketone body synthesis. Totally 8 to 10 cycles. You have to be thorough, doctor. So if you look at various steps, glucose to glucose 6-phosphate is hexokinase, which is reversible because currency is used. Glucose 6-phosphate to fructose 6-phosphate is reversible, right? If you became assistant professor to associate professor, you don't want, you can come back also, because there is no money involved in that. But you became director of medical education. There is an investment initially, then after that there is a return. So that is the reason it become irreversible. So पैसे की मामला होता है, जहां भी ATP का खर्च होगा. Similarly, doctor, the various steps, fructose 6 to 1,6-diphosphate, पैसे की खर्च होगा. तो इसलिए ATP is exhausted, it is irreversible. So you must know phosphofructokinase, pyruvate kinase, hexokinase. Whenever kinase is involved, phosphate is involved, it is irreversible step, is what you have to ultimately remember. What are high energy phosphate compounds is my question to all of you. ATP, creatine, phosphate are called high energy phosphates. Then urea cycle. Some part of urea cycle occur in mitochondria, some part occur in cytoplasm. You must know all the steps. So doctor, out of all this, glutamate dehydrogenase, alpha ketoglutarate dehydrogenase, isocitrate dehydrogenase and fumarase, they are all the ones which are involved in, which are not involved in urea cycle. Only arginosuccinate synthetase is involved. Okay, let us see. There is arginosuccinate synthetase is involved. Let us see. Arginosuccinate forms from the citrulline, and during this process one aspartate is basically released, and in that step you have arginosuccinate synthetase which is involved. And where is it located, doctor? Cytoplasm. Arginosuccinate loses one fumarate. Ultimately इन सब की आत्मा कहाँ जाएगा? Aspartate, fumarate, इन सब की आत्मा citric acid cycle में चले जाके स्वर्ग द्वार से इंद्रलोक को चले जाएगा. So you must know what is the fate of these end products which keep coming out. Then arginine causes ornithine to form, and arginase is involved. That is also cytoplasmic. So summarize what are cytoplasmic enzymes in urea cycle, doctor? Arginase, arginosuccinate lyase, arginosuccinate synthetase are basically cytoplasmic. Whereas carbamyl phosphate synthetase 1 होता है यूरिया साइकिल में, क्योंकि 1 फिंगर होता है यूरिन चलने के लिए क्लास में. तो इन स्कूल वी यूज्ड टू से टीचर 1 little finger, 1 finger means यूरिया. सो 2 होता है carbamyl phosphate synthetase, 2 is involved in pyrimidine synthesis is what you need to remember. Then ornithine transcarbamylase also is basically located in the mitochondria. Better to remember these two, located in mitochondria, is what I want to underscore to all of you. Now doctor, protein separation, which year paper? 2012 May paper we are with. Protein separation based on molecular weight कहाँ होता है? Protein separation based on electrical charges कहाँ होता है? Electrophoresis, doctor, it looks like a stupid topic in biochemistry, but you take previous aims, PGI, All India, any paper, they keep asking what is SDS page technique, what is polyacrylamide gel electrophoresis. Those things which we hate to read will become the high yield topics in biochemistry, but still you should एक अच्छा ईरानी चाय पी के एक बन खा के बैठ के पढ़ने वाला टॉपिक्स होता है इलेक्ट्रोफोरेसिस. You have to read and go to the exam, right? So native gel electrophoresis, 2D gel electrophoresis, ये दोनों में इलेक्ट्रिसिटी के वजह से सेपरेशन होता है, मगर अल्ट्रा फिल्ट्रेशन, जेल फिल्ट्रेशन एंड अल्ट्रा सेंट्रिफिकेशन, ये तीनों चीजों में because of the molecular size the separation of proteins occur is what you need to remember. What is not a transport protein out of all this? The very name says transferrin is transport, celluloplasmin carries copper, hemoglobin carries iron and oxygen, albumin carries lot of things. So that's what you need to remember. What will selenium deficiency will lead to? Keshan's cardiomyopathy, selenium deficiency. Once more, एक हैदराबाद ईरानी चाय पी के याद रखने वाला टॉपिक होता है ट्रेस एलिमेंट्स. एक टेबल रहता, 4 or 5 trace elements, their deficiency features, right? Hypogonadism, where is it seen? Dermatitis, where is it seen? Immunodeficiency is due to copper or magnesium or manganese or silicon, or the list of entrants is unending, doctor. Copper containing enzymes क्या होता है? Basically superoxide dismutase, cytochrome oxidase, tyrosinase, amine oxidase, they're all basically the copper containing enzymes is what you have to basically remember. Chaperones is another high yield area in biochemistry. Chaperones are also called heat shock proteins. Basically, they have got a lot of, they are pleiotropic in their properties. They have got wide range of expression. So how did Darwin thought that we are evolved from lower species? Looking at monkey's face, he discovered humans are similar. But later on there are proof that bacteria have same things which we are also having. Right? So bacteria and human, what is common thing is chaperones. And they are also called stress proteins is what you need to basically remember. Ubiquitin is not a chaperone. And most of the chaperones, they have a ATPase activity. And why chaperones are needed? For a proper folding of the proteins, we require chaperones. Any improper folding of the proteins will lead to prion disorders, which are neurodegenerative in nature, as all of you know very well. Then what is ubiquitin? What is its function? Ubiquitin is a very, very important question. Ubiquitin has a very important role in degradation of the proteins by the proteosomes. Coenzyme Q is there, no, in electron transport chain. So it is also related to ubiquitin is what you need to remember. Then proteomics study, what are the techniques which are used for knowing the protein expression in proteomics? Honestly, to become a interventional cardiologist, you don't need to know all this crap. But if you take the previous AIMS PGI, I don't know why, proteomics is a repeatedly asked question. Wikipedia writes a good article on proteomics, right? So morning, if you don't get a newspaper, at least while having coffee, you must pick up few most disinteresting articles, topics which are high yield topics like this, you need to read at the coffee table, right? So gene array chips, mass spectroscopy, 2-dimensional electrophoresis, they all basically are used in proteomics. I leave that for you to review on Wikipedia on proteomics. Now doctor, restriction endonuclease. RFLP is a very, very state-of-art molecular technology to decipher the protein structure, genetic structure, etc. So palindromic sequences are basically observed. For example, if you have a normal allele of a gene, abnormal allele of another of the same gene, the protein produced from a normal allele versus the abnormal allele, they will not be same in their length. They will have a length disproportion. Identifying the disproportion of the end product called the protein, we will identify the abnormality in the gene for which we use endonucleases to cut a particular allele and then search what is the product produced and how is that different compared to that of the original gene. So that is a restriction fragment length polymorphism with which you identify the mutations. So the restriction endonuclease is also naturally produced within the virus. So they protect the bacteria from the infection by virus. They are naturally also produced by the bacteria, and what is their purpose? Their main purpose is to restrict the replication of the DNA in the lower species is what I want to underscore to all of you. And scientists discovered these endonucleases which are normally used by the prokaryotes to restrict the multiplication and used it for the diagnostic purpose to identify the allelic mutations in the technique of RFLP is what need to be understood. What are the non-coding RNAs? I told you no, recent, is this question over? Last time we discussed some of the questions of PGI May 2012, but let me tell you doctor, we only know tRNA, mRNA, rRNA, but there are some Ajib RNAs which are the favorite area for the examiner. So the siRNA, miRNA and tRNA, rRNA, they are all called as non-coding because mRNA is the only coding RNA, simple common sense. But still let me tell you, RNAs are each of the function of this type of RNAs is a favorite area. I think recent PGI paper only we have discussed different types of RNAs if I recall remotely. Now because in actual exam if they ask about siRNA, miRNA के ऊपर क्वेश्चंस बहुत पूछ रहे जिपमर में एक्सेट्रा you must know at least 1-2 functions of each of the atypical RNAs. Now what are the techniques which are used to identify the mutations in the genes? Yeah, very good. So RT-PCR, then gel electrophoresis, DNA sequencing, RFLP, single strand conformational polymorphism. There are the various ways by which gene mutation are basically identified. So that's no big deal. Now about the histone protein. What is a true statement about it? Histone proteins are basically present inside the nucleus. See, DNA is there, no? To maintain the integrity of the DNA, you must wind it around something, and that something is histone, and it is basically basic in nature. About histone, 5-6 points commonly asked in the entrance. What are the two amino acids very rich in this protein called as histone, that are arginine and histidine? And they form the conjugated protein with the nucleic acid, that is the DNA, and they do not readily coagulate on heating, and they are synthesized in the cytoplasm but ultimately migrate into the nucleus. And there are modifications which occur within the histone proteins. What type of modifications? They are acetylation and methylation, and there is a significance for those modifications is what I want to underscore to all of you.